only 9 hours.
I'm certainly going to.
You gonna make it?
Yeah. We'll have to record. We'll have to record the entire private part of the defense too, and then see the questions they ask.
That would be awesome. Can you do that?
I'm sure you have a lot of stuff.
I love it like that.
A lot of things that you still want to put in.
Yeah, plates. Like real small.
Oh yeah.
I was maybe Friday doing this, but it's great.
Oh, but this is this is Hava. We and we were colleagues.
Right.
So we used to postdoc over at ING.
So you know
I don't think anyone actually understands research.
We'll find out. Maybe.
Yeah.
that's what I'm here for.
Which doesn't sound like a lot, but week after week, I kind of I feel my social skills atrophying. I didn't think
How are you?
I'm really good. How are you, Clarence?
Forever a week if you're working. Yeah, I mean,
That's what I'm finding. It's recording. We got a really good close-up of you approaching the camera.
Oh, is that the camera playing now?
Yeah, it's recording. Now's the time to say something snide.
Did it record me giving him that crumpled up piece of paper?
No, we can reenact the scene.
What is the crumpled paper?
I had a turkey, had a chunk of turkey too big a chunk of turkey for lunch, and so I might get a little sleepy. So that's if I'm dozing off, he throws that at me.
Brilliant. You planned ahead. This is so generous of you.
Because it's embarrassing if I did this to table or something. I get a bloody nose right in the middle of the fence.
Or a face full of bread.
Brie cheese though, that's looks like really good brie.
Best that surgeons has to offer.
What?
Best that surgeons has to offer. I came this close to bringing wine.
You can do that with a permit.
Well, I decided that wasn't the right vibe for the defense. Wasn't the right vibe for the right hand. I want half the audience.
But that's been a lot to stabilize.
The air from the southwest.
It's been really high strung and really a challenge. I haven't seen him actively freaking out. Well, he has a few free practice, but I mean, it's been
Again, you can take something forward with it, so anything that I can still do, it'll be like, it'll be fine.
Three.
Like, you do the strategy with like a public seminar.
Nobody wants to say.
Well, I still do this. When I was here, when I was in grade school, I used to. Always said
Is this box? That's what the paper was for. We could use that for
Who's this?
Yeah, like time
Thank you.
You recording this?
Yes, we are. Posterity.
You saw me that disagree with them. I couldn't even get angry with them.
Yeah, I agree.
It's good though, because I think that gives us an opening to put back in. We aren't right now. It's perfect. We love our reviewers very much. They're just wonderful.
You should
I'm like that for a while, so I apologize. Don't mind me, eye contact.
Yeah, the tripod doesn't look like it rotates that way.
You got to disappear? Just a little distance. Well, thank you all for coming. I'm not surprised, but I'm totally delighted by how many people came out during the last 2 weeks when most people who worked too hard all summer decided to take a vacation. So, well, I've been vacationing all summer, so I had to be here. Well, it's my pleasure. Adam has been with us for 5 years and 363 days. Some people take 6 years, but Adam wouldn't do that. And you just heard something that we'll miss, his laugh. Adam's laugh is something that keeps our floor smiling all the time.
And unproductive.
Well, unproductive, let me see. Since you've been here, not counting anything in your thesis, you've published 5 papers. So you didn't keep, it might have done it to us, but not to you. So 5 papers. I love the journals, Ecology, AmNat, Journal of Ecology, and Agronomy. There you go. That's pretty diverse. I like it. Well, lots of things. There are lots of things I like about Adam, as all of you know. He has a very broad range of skills. He's excellent in mathematics and theory, wonderful in stats. I always like to kick a grad student out around the time they realize they know a lot more than I know. It's time for Adam to go. So let me see what else. He's going from here to IDIV, which is this International Biodiversity Consortium formed in Germany. I think you said in Leipzig. He's also in Jena and someplace else. Halle, yeah. A good bread. Anyway, but the fun thing for Adam is going to be that he speaks Austrian German, because his mother's Austrian. And the Germans don't know how to understand this. So interesting to see what happens. You'd be better off, I advise you to speak English. Okay, anyway, with all of that said, I had a lot of notes here. Kathy told me I could not let him pass, that's my wife, because she would miss him too much. So I have to ignore that and give an unprejudiced judgment of the thesis today. So with that, we'll hear from Adam. Here's his title, Constraints and Trade-offs. I've seen that phrase before. And the rest.
Hello! Thank you all for coming.
Sorry. And there's a party at my house tonight at 7. And if you want to come, let me know, and I'll tell you where it is.
Whether or not he passes.
Yeah, really.
Absolutely.
There'll be a party or a wake. So, hi everybody, thank you for coming. As Dave said, the title is somewhat familiar to many of you. This is actually sort of robbed from a paper that Dave put out in the 1990s. And in a lot of ways is a continuation of a lot of that work. And so the main goals are going to be to see if we can find a couple of ways to introduce some theory from trade-offs into models of community assembly to try to better understand how different groups of species come together and what the consequences of those are for ecosystems. To give you a quick idea of the structure we're going to follow, I'll have a quick introduction, and I'm going to walk through the thesis chapters that were part of my dissertation. The first is going to walk through some pretty classical methods for analyzing ecological data based on pattern matching. So basically fitting model parameters until you get a model to look like what's happening in a system that you're trying to mimic. Chapter 2, we're going to talk about some potential problems with those methods and introduce some ways that you might go about solving those problems. Chapter 3, we're going to move into some theoretical discussions about trade-offs and how those might help us find low-dimensional approximations for the important mechanisms that are driving the system. And then in Chapter 4, we're going to go over some ways to apply those to real-world systems. The way that you're going to know where we are in this talk is both there's going to be a status bar at the bottom that we'll just move through, and also come back to this slide every time we transition between sections. To begin with, though, I wanted to start with a picture from Cedar Creek. So Cedar Creek is an ecological reserve about 40 miles north of where we are right now. A lot of the data that I'm going to be talking about came from this site. I particularly like this photo, both because it shows Cedar Bog Lake, which is a big piece of research at Cedar Creek, and also because it shows it from an angle we don't usually see. This is coming in from the southwest, from a plane that's basically sitting right on top of the creek itself. So the creek is at the bottom of this picture. And it does a really nice job showing off the different kinds of ecosystems that we have at Cedar Creek. Up in the back, you see prairie, sort of mixed throughout this picture. You see mixed deciduous forest. There are a few patches of white pine, which is reasonably rare for this area because so much of it was logged out. And then as you head in closer towards the river, towards the creek, you see things like tamarack, which is a water-loving tree. And then as you get really into the marshy area, you get these emergent plants like willow and reed canary grass. And if we were to pop up another scale, we would see similar patterns. So around Cedar Creek, for example, we know there's smaller scale heterogeneity. But in general around here, most of the communities you run into, most of the mixes of species, will be something corresponding to a deciduous forest. And if we went north, we would move into something more like a coniferous or boreal forest. Those are different. And if we move west, we'd run into prairie or parkland. And similarly, up at the scale of the globe, we know we would run into different kinds of mixes of species here on the eastern half of North America versus if we went into Southeast Asia or Southern Africa or something like this. And what this all goes to show is that different species seem to be found in different places. And that's largely because the conditions under which individual species can persist are limited. There don't seem to be a whole lot of individual species that are able to persist everywhere. And sort of going along with that is the notion that since these species seem to have distinct ways of interacting with their environment, they also must have distinct effects on one another and on the systems that they live in. A big sort of contention that we're going to be working from for most of this talk is going to be this notion that because different species do different things, communities that have lots of species in them tend to be able to do more things than communities that have a small number of species in them. And since humans tend to rely on a diverse mix of services, this would suggest it might be important to understand how different types of communities provide different sorts of services. Now, in most talks, here's where I would go in to show some of the quantitative data that we have from places like Cedar Creek, showing specific links between particular types of services and both general diversity and the diversity of particular types of species. But instead, I thought I would show some pretty pictures of some diverse systems as sort of a proof of concept for the sorts of things we get out of these. So to begin with, here's a picture from Isle Royale with a lady slipper in the front. These are the sort of ecosystems that people around here like to just go out and experience, specifically because they're not particularly heavily managed. We just like seeing the sorts of things that arrive there on their own. We also like to construct ecosystems, right? So here's a picture from the Palm Room at the Como Conservatory. It includes species from South Africa and Eastern Asia and South America, all sort of thrown together in one room. And so there's no place on Earth that looks like this in a natural system, but we assemble it like this because it's sort of aesthetically pleasing. And so people are willing to put in resources to maintain a spot like this and pay money to go visit it. Similarly, for more utilitarian uses, this is a green wall from the Museum of Science in Boston. And this is a nice proof of concept, both as an example of social benefits. There's a lot of research showing that having something like this in your building just makes people happier, but also sort of utilitarian stuff. This shows how you might use a system like this, both to clean water in a building and also clean air. A system that's particularly near to my heart is this. These are the Alps in southwestern Styria, where my mother's family is from. And this is sort of one of the primary cultural and tourist-based pieces of that region. These nice open meadowy mountains, which you can go hiking in. There are these systems of huts where you can go from hut to hut and enjoy them. And what makes these systems particularly interesting is that these open spaces aren't a result of climate, they're a result of management. So these are only at 1,000 to 2,000 meters, meaning if they weren't grazed, as most of them are, they would be forested in pretty quickly. And so not only does grazing these systems provide a nice backdrop for your touristic adventures in Austria, but but there are also some other side effects that we get. So we get beef from these systems, we get dairy products, and because we maintain forest in the lower regions, we also get a nice source of mushrooms and blueberries to round out your meal. So this is a nice example of a nice mixed system full of lots of different things that we can get out of it. Of course, the world is always complicated, and there are always some side effects to running something like this. So this is a nice low-yield system where you can have employment opportunities and nice aesthetic values. But we also know from all sorts of research, including recent stuff put out by Dave and other folks in the Tillman lab, that if we were to pick the things that are most endangering species around the world, it would be low yields, not being willing to subsidize areas that only have low yields with food that you can grow in another region, and also insisting on diets that are high in meat products, like the cattle that we use to keep our systems open and productive in Austria. And so all of this just goes to show that even though we get lots of things that we like out of ecosystems, often they're not quite as simple as you might think at first blush. And so really understanding the broader implications of how you design a community and what you get out of it are pretty important. And so moving from there, we're going to have sort of three main overarching questions that I'm going to focus on in this talk. The first is whether we can understand the causes that make different sorts of communities assemble in different places. Why is it that we get one type of mix of species here and another type of mix of species here? Second, we'd like to know if we can predict what kinds of species and sort of properties of an ecosystem we would expect to develop in one place or another, given some set of things we can measure about that site. And then third, the real sort of holy grail here would be if we could find ways to alter those ecosystems to do things that we would like them to do. So sort of figure out why things are put together the way they are and whether we can mess with them in ways that change them in ways that we'd like them to be changed. So to begin with, I'm going to jump into a pretty classic pattern matching analysis. Like I said, by pattern matching, I mean fitting a model to mimic a pattern that we see in the real world. And for this, we're going to focus on succession in old fields at Cedar Creek. Now, succession describes the naturally occurring transitions in species and environmental conditions that you see following a disturbance event. And old fields in particular are systems that are undergoing succession following some kind of active management after that management has abandoned. So typically has been abandoned. So typically the term is used to apply to areas that used to be under agriculture or pasture use. And here we're going to be focusing on areas that were previously used for agriculture. Old fields are important in a lot of ways in ecology. They're one of the systems that was used for a lot of the foundational theory that we use in community assembly and community ecology. They also make up a reasonable portion of total land area. So a lot of the world is currently being used as pasture or cropland. And in places like the United States, something like 10% of the continental United States is actually cropland or pasture that has been abandoned within the last 100 or 200 years. So understanding how these systems work is of some sort of broader utility to us. The general pattern of old field succession is usually something like this. You'd have some kind of a disturbance for naturally occurring succession. It might be something like a fire. For old field succession driven by a change in management, it might be something like slash and burn agriculture. Once the disturbance ends, once you stop tilling your field, for example, you then tend to get a pretty regular series of species coming in. You first get invasion by weedy annual species. These eventually get replaced by longer-lived herbaceous perennial plants. And then finally, in an area like this around Cedar Creek, in general, it's thought that the long-term fate of these systems is some kind of a forest, some sort of a deciduous forest in this area. Cedar Creek is a great place to study old field succession because it has a number of long-term studies in the region. There are 24 fields which are now part of long-term surveys in the area that were set up by Dave in the early 1980s. And because of that, we have two kinds of data in these fields. We first have the long-term records that we have from repeated surveys that have happened within individual fields since the 1980s. But we also have what's known as a chronosequence, and that's because these fields were abandoned at different periods. Some were abandoned as early as the 1920s, and others were abandoned as late as 1 or 2 years ago. And so we can compare across these different types of fields, these different ages, as it were, to get a sense of what a very old field looks like versus a recently abandoned field. The other thing that's special about this experiment, and that makes it an experiment, is that we have, for the past 10 years, half of the plots in each field have been burned experimentally at a frequency of about once every 2 to 3 years to test the effects of repeated disturbance on the system. The general sort of average trends in these fields are pretty well understood by now. There are a series of studies by David Inouye and Dave Tillman in the late 80s and early 90s. And so typically near the beginning of succession, you see things like these species up here. So weedy annuals like ragweed or horseweed. After sort of 2 to 3 years, you start to get invasion with sort of mid-successional species, which are usually C3 or cool season grasses, things like quackgrass or Kentucky bluegrass, which would be the grass that you probably seed in your lawn. And then late in succession, around year 40 or 50 or even 60, you usually get invasion by sort of these late successional dominant bunch grasses. So things like the C4 grasses, little blue stem or big blue stem, which are these long-lived perennial species. And in general, as one of these later successional groups comes in, they tend to push out the species that had been there previously. There are 2 other groups that I'm going to be focusing on as well. These are perennial forbs and perennial legumes. They both tend to be present at some low abundance throughout succession, and they follow somewhat more variable patterns in terms of how they progress. Now, if we look at the individual trends from this long-term data set that we have now, here each black squiggly line is the trend from a single field through time, and the age just shows how many years it's been since that field was abandoned. You'll notice that for some groups like annuals, there's a pretty obvious trend, which is things start out common and then through time they become rare. But for other groups, it's a little bit less clear what kinds of trends they follow. In some cases, there are a couple of similar trends, but with some variability around it, and in others, it's sort of unclear whether there is a trend at all. And so the main question that I'm going to be focusing on in this section is to see if we can figure out if there are some major drivers that cause this variability among fields. And in particular, we want to know whether there's sort of a single pattern that these fields follow with a little bit of variability around it, or is it actually that there are fundamentally different kinds of succession that are happening in different fields. And regardless of which of those 2 things is driving variation, we'd also like to know if we can do something to predict the kind of succession that we'll see in a field given some set of measurements we take. The way we're going to do this is to focus on drivers and responses. So the drivers we're going to focus on are soil fertility, the burning treatments that I talked about, long-term climate, so long-term averages of precipitation and temperature, and the effects of competitive interactions. And we're going to focus on those because they've been identified in previous studies at Cedar Creek of being particularly influential on plant communities in the area. There are a few variables, things like herbivory, that are certainly important at Cedar Creek, but that we're not considering because we don't have a very good way to distinguish differences among the fields for those sorts of things. So these are the things that are influential and that we have good data for. And we're going to focus on how these things are associated with 3 types of responses in old fields. The first is the abundance of species in fields where they're present. The second is going to be dispersal of species into fields where they're not yet present. And then the third will be mortality within a field where they were present. And so dispersal and mortality are effectively a species arriving or disappearing from an area over the course of the survey. And what we're going to do is we're going to use some pretty standard regression tools to link these drivers to these response variables. And then we're going to link the response variables together using something called a metapopulation model. In this case, it's going to be a very simple simulation. It just tracks abundances across lots of different sites to see how interactions across those sites lead to sort of larger scale meta dynamics. And then once we have this integrated model that includes both the drivers and the responses, we're going to use that model to test for potential differences in successional trajectories related to these drivers. So for those of you who are into this kind of thing, these are sort of rough ideas of what these models look like. For the abundances, we're using what's basically a pretty simple linear regression with some fancy magic to account for nonlinear effects of age. And basically, we just have individual terms for each of these drivers that I mentioned, and we use a regression method just to find estimates for all of these beta terms that give us the best predictions of abundance given the predictor variables that we have. For colonization mortality, the setup is quite similar. Again, a bunch of linear predictors, but instead of a standard linear model, we're using a binomial model in order to predict probabilities of colonization mortality events. We're then going to stick these together in a really simple metapopulation model. Basically, what we say is plots are either occupied or not occupied. That's what our p variable tells us. If a plot is occupied, by a particular species group. We assume that that species group takes on the abundance predicted from our abundance regression. If it's not occupied, we assume the species group has an abundance of 0. And then we decide whether or not plots are occupied using the probabilities of colonization mortality that we can get from those two mortality and colonization regressions. So this is sort of the stuff we're sticking together, and what we get out of it is something like this. So here are the results from that model for the average conditions in unburned fields. And what we've done is just simulated dynamics across 90 years of succession in these systems. The shaded regions here show a mean plus or minus 1 standard deviation across simulations. The solid lines show the average trends observed across all fields, and these gray dashed lines are showing trends for individual fields. And so you notice in general we capture some patterns that seem to more or less match what we had seen in these earlier studies. So in general, C4 grasses, these late successional species, start out rare and become abundant. C3 grasses, the mid-successional species, sort of peak in abundance and drop out. Annuals, the early successional species, start out as abundant and then fade away. And forbs and legumes follow sort of intermediate patterns, but in general stay rare throughout succession. Now what we can do is look at how different changes in the drivers lead to variation around this mean result. And that's what this next figure is showing. This is called an effect size figure. And this is measuring fold change, which means for each one of these figures, numbers like a positive number 1 would mean that we find twice as much biomass in these plots as we would have found under the mean conditions for some time period in a group. A value of negative 1 would mean we find 100% less biomass, so the species is absent. Again, the shaded regions here, and I'm going to go through these one at a time in a minute. The shaded regions here correspond to results from the simulations. The solid and dashed lines are showing you observed variability within the fields. They're not expected to line up perfectly because the simulations are testing events that are much more extreme than we've seen in the real data. So the top is showing results for a field that has been burned since it was abandoned. The middle is showing results for plots that are as fertile as the 95th percentile of most fertile plots observed in fields. And the drought simulation shows conditions for dry, hot years that correspond to the long-term projection of what we would expect Minnesota's climate to be like around 2050. Now, even though we can't necessarily test the predictions against observations in the fields, we can get some insight by comparing them to experimental results where we've manipulated things a bit more broadly. So for example, if we compare our results to burning experiments at Cedar Creek, in general we find that when we burn frequently, we increase C4 abundance and decrease forb abundance. And indeed, that's what we find here. We find higher C4 abundance than we had for the average simulations and higher forb abundance. We know from nitrogen addition experiments at Cedar Creek that in general, higher rates of nitrogen addition into systems tend to be associated with increases in C3 abundance and decreases in the abundance of other species. Again, that's what we find here. So increase in C3 grasses, no increase in the other species, and a decrease in things like annuals and forbs. And then finally, from observations of intense droughts, the most intense drought that we've had in the last 50 years, in general, we find that most species groups become less abundant over the course of a drought, but that C4 grasses are a bit more resilient to those effects. And indeed, again, that's what we find. We find that all groups are predicted to be less abundant in these drought scenarios, but that the C4 grasses are the least affected, and in fact, by the end of succession, are effectively not affected anymore within this simulation. So, you'd think from all of this that these drivers must have some pretty profound effects on the successional trajectories that we see in the old fields. But that turns out not to be the case. So here, these figures are the same as what we just looked at before, but instead of looking at differences from the mean simulation, this is just showing the time trend in each field. So this is just showing the relative abundance of each species group as a function of time. And what you notice is that they all pretty much follow the same trend. So the purple annuals start out common and decrease, the teal C4s start out rare and increase, and the orange C3s sort of have this U-shaped curve. And what this goes to show is that even though there are detectable and significant associations among the drivers and these response variables that we measured, it seems that they don't seem to have a big effect on the overall shape of successional trajectories. And so this would suggest that even though there's some variability in what these trajectories look like, that variability isn't big enough to cause fundamentally different kinds of succession. We're basically seeing variation on a theme. So this would be consistent with the expectations of having a single type of succession with some wiggling around it, driven by environmental variables. One potential reason for why we might see this would be some compensatory trade-offs within this system. And I have to admit, I'm a little bit embarrassed to show this, since this is a regression of only 5 data points, since we only have 5 species groups. And we'll see in a minute why we're using so few. But in general, what we find is that there's pretty strong associations between the mean age response, which is a sort of an indicator of what we got from our abundance regression, and the mean colonization and mortality rates predicted from the 2 binomial regressions. And what these are basically showing, age response is a pretty good indicator of the competitive hierarchy of these species. So species that have a negative response to age, things like annuals, are things that are getting displaced over the course of succession, whereas things that have a positive response to age tend to be things that are coming in over the course of succession and displacing other species. And so we find that here, the groups that this index would say are good competitors, the ones with positive responses to age, also tend to be bad colonizers and have higher mortality rates. And again, I'm not sure how seriously we'd want to take these results, given how few observations we have to fill these spaces. But it would suggest that one of the reasons we're not seeing a lot of variability across successional trajectories is that if we change conditions in a way that does something like favor good competitors or favor good dispersers, there are going to be some trade-offs within the system that sort of dampen out those effects. And so in general, we're going to be held towards that mean trend that we see. So, sort of summarizing what we have here, we've seen that we can find these significant associations among the drivers that we considered and these response variables of abundance, mortality, and colonization, and that while their combined effects do seem to be associated with these shifts in successional trajectories that alter things in detectable ways, they don't seem to lead to divergence in successional trajectories. And lastly, an explanation for that might be these compensatory interactions within these systems. So, with that background, we're now going to move into some potential problems with that pattern matching approach and some possible solutions for how we might address those patterns. The most obvious problem with the previous approach is predictive power. So here I'm showing the R-squared, so the fraction of observed variation explained, just from the abundance regressions. And so remember, the abundance regressions were a pretty complicated model. We had a lot of terms that we were using to make predictions. But in general, our predictive abilities aren't that high. We're never explaining more than about 50% of the variation that we see in the data. And more importantly, as we go down to rarer species, things like forbs and legumes, we basically lose explanatory power. We're not very good at explaining legume dynamics at all within this model. And so within this section, we're going to be focusing on trying to figure out some reasons that we might expect to see these poor fits from a pattern matching approach like what we just used. And we'll talk about some solutions that might help address these problems. And we're going to focus primarily on interactions among species. So that's really one of the more important drivers of outcomes in competitive systems. To do this, we're going to focus on a model that was posited by Robert MacArthur in the 1970s. It's a sort of simple toy model that posits a possible mechanism for resource competition. And we're going to use it because it's a quite simple model, but we'll talk a bit later about some potential problems with it. So in this model, we imagine that there are some set of resources that are all perfectly substitutable. And what that means is that every species in this system should theoretically be able to survive on a sufficient quantity of any one of those resources. And so one good example of this sort of a resource might be food items that all have the same nutritious value but are different sizes. And species within these systems differ in their abilities to acquire each of these resources. So you might imagine, for example, you have birds with different sizes of beaks, and birds with big beaks are good at eating big seeds, and birds with little beaks are good at eating little seeds, something like that. The guts of this model are pretty simple. Each species has a series of consumption parameters, these CIL terms, where I is an index for the species and L is an index for the resource. And so if a species has a high CIL, that means that species I is efficient at consuming resource L. In this model, species only interact through resources, so species I only has effects on species J because I is eating something that J could have eaten. And because it's and we've simplified this model a little bit from the one initially posited by MacArthur, But in the version we're using, you can estimate the effect that species have on one another. This is called the competition coefficient or the interaction coefficient, simply as the sum of the products of these consumption parameters across species. So the effect of species J on species I, that would be Aij, is just the sum across all resources of CIL times CJL. Now, because we've posited a particular mechanism, a particular sort of plausible set of things that could lead to competition in this model, we've also inserted some pretty specific structure. And in order to demonstrate that, here's an example where we have 3 species, I, J, and K, competing for just one type of resource, resource L. And you'll notice if I color in these coefficients, there are a lot of shared pieces across these terms. So most obviously in this model, you notice that Aij and Aji, so the effect of I on J and the effect of J on I, are actually the same thing. They're the same parameter, just in a different order. But even if we compare other terms like Aij and Aik, you'll notice that they still share some parameters. So CIL is in both of those. And what that means is that there's some link among these parameters. They're not totally independent from one another. Said differently, because we have an underlying mechanism in this system, we've forced some links into the parameters that we're trying to use to explain things. Where this is going to manifest is if there's any variability in the system. So imagine, for example, within a single species of bird, there might be some variability in beak size. Not every bird of a particular species has a beak that's exactly the same size. So there'll maybe be a mean beak size with some wiggle room around it. And this variability, if our interaction coefficients are linked, is going to lead to something like this. So here, for example, are different observations that we might take of the effect of species J on species I. This is Aij versus the effect of species K on species I. And you'll notice that when one is large, the other is large, and when one is small, the other is small. There's covariance between these parameters. They're correlated because of this mechanistic structure in the model. This is particularly problematic for empirical studies because especially for things like competition coefficients, we tend to estimate those one at a time. So the easiest way to do it is often to grow 2 species together in lots of different plots. And what that means is if I estimate Aik and Aij in 2 different plots, I'll never be able to notice the fact that the 2 of these tend to wiggle back and forth in synchrony. So I'm going to miss the fact that there's a link between these parameters. And because of that, I'm likely to make some pretty big errors, both in terms of bias and in terms of accuracy, if I try to use these sort of simple, uncorrelated parameter estimates. So, to sort of demonstrate this, we're going to walk through a couple of steps. First, we're going to parameterize a bunch of versions of MacArthur's model that we just described with some variation, some process noise in the consumption parameters of individual species. Second, we're actually going to do a little bit of fancy math. And I should say by we, I mean Claudia did a bit of fancy math with which you can extract the expected magnitude of the variation among parameters within this model. So we can actually look at the actual mathematical structure of this model and pull from that the expected size of correlation among the different parameters. And then we're going to simulate 2 different versions of the model. We're going to simulate one where we don't account for correlation among parameters, so an uncorrelated model, and then a second where we do account for these covariances among model parameters. Just to give you an idea of what these look like, depending on the particular combination of interaction terms, if it's Aij versus Aji or Aij versus Aik, there are slightly different expected covariance terms, but they basically just depend on the mean interaction values and on this term sigma c, which is the magnitude of variability that we see within individual traits. And so as a simple example, here would be the predictions we would make of 2 species competing with each other, where the vertical axis shows the probability that one species competitively excludes the other. The black line here shows the actual results we would get from the true mechanistic model if we simulated that. The red result shows what happens if we make predictions using the uncorrelated model. And so you'll notice particularly as that variability gets large, as sigma c gets big, our ability to predict this system declines. And in general, we start to over-predict instances of competitive displacement. Whereas if we correct our estimates using this covariance structure, we can actually do a pretty good job of approximating the correct amount of competitive displacement within this system. Expanding this out a little bit, we can simulate this for species with between 1 and 5, for communities with between 1 and 5 species and 1 and 5 living resources. And what we find are indeed these strong biases and changes in precision associated with these methods. So for these, we're showing estimated richness and abundances of species in these communities. The red shaded region shows mean simulation results plus or minus 1 standard deviation for the uncorrelated model. The blue shaded region shows the covariance model, and then where it's purple, that just overlaps between the two distributions. And of course, the black line is the true value from the mechanistic model. And so what you'll notice here is first, there's bias in the estimate that we get from this uncorrelated model. And by bias, I mean that it's not centered on the correct value anymore. I mean, even if we could measure this really, really carefully, we in general would overestimate this number here. By precision, what I mean is the size of these confidence intervals. And so you'll notice that there's much bigger spread in these estimates attributable to the uncorrelated model than to the covariance model. And so this does a very good job of both showing the problems that come from not accounting for these mechanistic relationships among parameters and the degree to which we can solve that problem by incorporating a little bit of that relationship using these covariance estimates. So our summary for this section would be that models that include some specific mechanism of species interactions can lead to constrained relationships among parameters in our model. Pattern matching approaches can miss those associations, which can lead to poor predictions. And that finally, if we account for covariance among these patterns, so this estimate of relationships, we can actually improve some of those predictions by inserting some of those restrictions back into the model. But again, there are going to be some caveats to this approach. So first, like I said, the specific model we used is a bit problematic. If we were to sort of imagine a real system where this model applied, there would be a very strong selection pressure driving species to specialize in individual resources. And so we would be unlikely to see communities following this model in the real world. We could, of course, apply this sort of a method to more complex systems. That would be a totally, we could pick up this approach and do something similar elsewhere. But one problem is that covariance doesn't always include a lot of information. And so for more complicated models, there's a chance that even if we account for covariance, that wouldn't be enough to actually capture the relationships among variables. And more importantly, it could be that if we're trying to compare lots of complicated models that are similar to one another, we might not be able to distinguish these similar mechanisms using this kind of a method. So, with that in mind, we're going to move on to the third stage of the talk and talk a bit about theory that we can take out of insight from trade-offs and whether we might be able to use some of that to improve to help solve some of these problems that we've been encountering. So to begin with, I'm going to basically make an assertion and hope that you guys believe me. And this is pretty widely agreed upon, that if you look across the suite of models of species interactions that include plausible mechanisms, that is, that they're actually meant to mimic a process rather than just match a pattern, all of those models include some sort of a trade-off, and that prevents one species in the model from just being better at everything than all the other species in the model. And so because of this, we expect there to be trade-offs in any one of these mechanism-based models that we might be able to think of. And so based on that overarching idea, two questions we'd like to try to answer is whether trade-offs contain useful information about the mechanisms that are leading to species interactions within a particular system, and second, whether or not that information is actually useful. And I'll get to in a moment why we might have some doubts about that. To begin with, a quick walkthrough of what trade-offs are. A good sort of general definition of trade-offs is that they describe a series of strategies among which there are constraints, such that increasing investments in one strategy necessarily leads to decreasing investments in some other mix of strategies. So to get better at one thing, I have to get worse at some mix of other things. I'm going to parse this a bit more finely following a framework that Dave has developed over the last couple of years into both physiological and ecological trade-offs. And so physiological trade-offs broadly describe this sort of frontier that limits the kinds of strategies that species can take on. And so these limits are just based on sort of the fundamental physical and energetic limits of systems that keep you from investing, say, the same unit of energy both in your leaves and your roots at the same time. Ecological trade-offs sort of provide a backing to that frontier. And the insight behind these is that there might be lots of combinations of traits that are physically possible, but are just not advantageous to persisting in a particular system. And so even if these traits could potentially happen, you're very unlikely to see them in the real world because those species wouldn't be around anymore. They would have been pushed out by something else. To demonstrate these a bit more clearly, I'm going to use a resource competition model developed by Dave. And this has sort of become a bread and butter model for doing a lot of resource-based comparisons within diverse communities. In this model, we have two resources here, resource X and resource Y, and species in these models have traits corresponding to those resources, which we call R star. So R star X and R star Y here refer to the minimum quantities of those two resources that these species need in order to have positive growth. So for example, if there were an environment somewhere that had that much resource X and that much resource Y, the species would be able to persist because there's enough of both. Whereas in these two systems here, the species wouldn't be able to persist. In the top X there, because there's not enough resource X, and in the bottom X, because there's not enough resource Y. In general, in these models, it's helpful to think of resources along a gradient. So you might imagine this represents heterogeneity in the distribution of resources across space. And so again, here, the sites I've marked green, the species could potentially persist on its own, and in the sites I've marked red here, there aren't enough of one or the other resource for this species to persist. Skipping over a couple steps, within these systems you can also have coexistence among multiple species, the primary driver of which would be that they fall along a physiological trade-off. And so the specific regions of coexistence and competitive exclusion depend on the exact consumption parameters in this model. But in general, you end up with some region, if you have two species, where species B competitively excludes A, where A competitively excludes B, or where the two species coexist together. And what I mean here by a physiological trade-off is simply to say that in order to become a better competitor for resource X, so to decrease my R star X so that I can survive at lower resource concentrations, I need to give up competitive ability for resource Y. So this doesn't have to be a straight line, but it does need to be a monotonic function. Alrighty, and the nice thing about this model is provided that I have heterogeneity and that species follow along a trade-off, then I could in theory have an arbitrarily large number of species coexisting in this system. So there'd be regions here where species A, B, C, and D are able to persist. Like I said, the ecological trade-off in a lot of ways provides a backing to this. And so you might imagine, for example, if that line that I've marked here is the physiological trade-off, there could be these species 1, 2, 3, and 4 that fall away from that line, so they're just worse competitors for both resources than that frontier would suggest. But these species are likely to get squeezed out in most real-world communities because something that actually falls on the real trade-off surface, on that physiological trade-off, would be able to outcompete them in all environments. And so you would expect selection to drive species towards that physiological frontier. Now, this seems, are people with me so far? Are we happy? Okay, so, seems simple, good. The reason that we might have some worries here would be just the fundamental complexity of a biological organism. So this, for example, is a simplified diagram of the Calvin cycle. So this is a simplified version of some of the stuff that land plants do. And if we were to look through this, we could identify probably 100 things that correspond to things we might imagine are part of trade-offs. And so this sort of brings up the worry, if organisms really have thousands or tens of thousands of traits that might be part of high-dimensional trade-offs, do we really have a whole lot of hope that we can pull any useful insight from that, or is it just going to give us another high-dimensional system that we don't properly understand? I'm going to make the case that in fact we are going to find some low-dimensional utility in these systems. And to do that, I'm going to talk about dimensionality reduction in a competitive system. And so here we're going to start with simulated resource competition models, following the form that we just talked about, this Tillman resource competition model. We're then going to vary the number of limiting resources and the size of communities within these models. And we're then going to let these communities evolve through time to some sort of a stable configuration of traits in their environment. And then finally, we're going to test their responses to heavily adding one of the previously limiting resources, so heavily fertilizing with a resource that was previously limiting. And then we're going to test how that affects the distribution of species and traits in this model, and how it changes which sorts of species are able to invade a system. Now, the reason we're focusing on resource addition is that there's a lot of empirical and theoretical evidence suggesting that that's effectively the same thing as removing an axis from a trade-off. So here, as we move from A to H, for example, these are results from plots at Cedar Creek, where we move from A, which is unfertilized, out to H, which is heavily fertilized with nitrogen. And you notice through time, the plots that receive the highest fertilization tend to have the largest loss in species through time. And this has since been replicated in lots of different places. We have results for grasslands in California, grasslands around the world as part of the NatNet experiment, and algal systems. And so in general, if you heavily add one of the previously limiting resources in a system, you tend to find declining numbers of species, and in theory, you would expect this to result from the loss of one of the previous pieces of a trade-off that had allowed for coexistence. To sort of demonstrate that a bit better, on the left here is a physiological trade-off surface corresponding to a trade-off among three resources. So you can imagine this as being a plane that's sitting in a three-dimensional space. And species have traits that correspond to their competitive abilities for resource X, resource Y, and resource Z. You notice that if we now heavily fertilize with resource Z, such that it's no longer a limiting resource in this system, we drop to that lower dimensional system over on the right. And this leads to something interesting, which is even though these species were all on the same high-dimensional physiological trade-off surface, this three-dimensional surface, we find that this new system includes a lot of species that are far away from the two-dimensional trade-off frontier. So if we were to imagine the sets of traits that would make you the best competitor you could be under this new two-dimensional environment, a lot of species are pretty far away from that frontier. And that means that a lot of the species that were initially able to persist in that three-dimensional system are going to be driven extinct by species within this system. So for example, this species is able to draw down resources to sufficiently low concentrations that it might be able to wipe out all of these species here. The effect of that is going to be that this system sort of draws trait values towards that new two-dimensional trade-off. So if we were to measure the average traits in these systems before and after we've heavily fertilized with the previously limiting resource, we would both find that we've lost a lot of species, but also that the remaining species fall pretty closely to that two-dimensional frontier. More interestingly, if we were to leave this system for some long period of time after we've initiated this change, we would find that under almost any circumstances, we would expect that evolutionary selection would drive that community closer to that two-dimensional frontier, just because any change in traits that lost competitive ability for resource Z and gained competitive ability for the other two resources would result in an increase in your ability to grow in that system. Which means if we were to take a group of species that had evolved under this lower dimensional system and invaded them back into that three-dimensional system, not only would they fall close to that trade-off frontier, but they would also be able to exclude even more of those initial resident species. If we play this out across a larger number of species, we find a pretty similar pattern. So here on the left, for example, we show for communities of between 5 and 50 species, as we increase community size, a larger fraction of those resident communities get displaced by other resident species. So these are ones that just by chance happen to be near that new two-dimensional frontier. We find that the ability of invading species to come in and both become abundant and displace species tends to decline. And that's again because as these communities become more diverse, we expect species to pack closer and closer to that two-dimensional frontier just because there are more species filling up trait space. And sort of as you'd expect, the species that end up rare after that fertilization event or that get driven extinct after the fertilization event tend to be ones that have invested heavily in resource Z. That resource that's no longer limiting. And so if you've invested in that resource, it implies you've invested less heavily in acquiring the other resources. And so this is actually a pretty important implication because it would suggest not only that higher diversity communities seem to be better at clustering towards low-dimensional frontiers, but furthermore that both evolutionary and ecological selections, evolutionary selections that drive towards the frontier, ecological selection is just the difference between the species that make it and the species that don't. Both of these ought to push systems towards the lower dimensional trade-off frontiers that correspond to things that matter for a particular system. So even if there are thousands or tens of thousands of possible types of traits and trade-offs, there's some reason to believe that it might be that in a particular system, if we were to look across the traits of species that are able to coexist, relationships among those traits would follow some lower dimensional trade-off that we might be able to better understand. And so, with that piece of insight, we're going to move on to the last chapter here. And in this chapter, we're simply going to look at ways that we can apply that previous insight. So, given that trade-offs might contain low-dimensional information that we could use to identify mechanisms, we're going to ask how do we access that information? How can we apply that information to identifying important mechanisms? And then, lastly, whether we can use that the information to improve our predictions from mechanism-based models. To do that, we're going to focus on empirical trade-offs. So empirically observed relationships among species are pretty common. In this case, this isn't quite an empirical trade-off. The way that we're going to define these would be relationships among the traits of coexisting species. So these are relationships, points of particular species of plant somewhere in the world. An empirical trade-off would be if we took all of the points within this diagram here that corresponded to species coexisting in a particular spot at a particular time. So, as we've seen in some of the previous chapters, one of the main problem we're trying to overcome here is that these models that include mechanisms tend to be pretty difficult to parameterize if we just go out and measure the parameters of those systems in the field. That's things like the uncorrelated model that we discussed. So given that these empirically observed relationships among traits of coexisting species should include a little bit of information about both the physiological trade-offs that they follow along, so the things that are physically possible, and the ecological trade-offs, so the things that limit their ability to persist in a particular system. The thing we're going to test here is this notion that it should be possible to parameterize models with traits that have sort of been snapped to that observed empirical relationship among species. So we see some empirical relationship among the traits of coexisting species, and we sort of draw the parameter values of our model closer to that overall surface in hopes that we might sort of constrain measuring error or correlations among parameters or other things that might confound our model back towards the general trends that we see across lots of measurements. The way that we're going to do this is that we're going to first identify some traits here for plant communities at Cedar Creek that correspond to sort of that correspond to hypothesized mechanisms that we think might lead to coexistence in that system. We're going to use these traits to build a mechanism-based model around these potentially influential traits. And then we're going to test predictions in these models against observations. And so the important piece here is that we're going to measure traits in monocultures, so in individual species growing on their own. We're going to test those against observations in polycultures, so in mixtures of lots of species. And so this tends to be sort of good practice for a model, that I parameterize it using data from one place, I'm going to test it using data from another place. And critically, for these traits to be considered part of an empirical trade-off, we should hope that we can find some sort of an empirical relationship among those traits. And then if I were to pull information from that empirical pattern, I should be able to improve my predictive abilities within that system. We have a lot of data and insight to work with from Cedar Creek. We're going to be drawing from across these sets of experiments. And so specifically, we're going to pull information from whatever it is, 3, 6, 7 monoculture experiments. And so these include 247 plots with 35 different herbaceous grassland plant species. And then to test predictions for our model, we're going to use a series of multi-species mixtures, which include 122 plots, which have been planted with mixtures of 2, 4, 8, or 16 species. The last bit of insight we can draw on is ideas that have sort of theoretically been considered as important for coexistence at Cedar Creek. One thing that has been positive for quite some time is this notion that species that are very good competitors, and so here this would be the R star, as we talked about in the previous model, for soil nitrate, which is a good indicator of species competitive hierarchies for nitrogen. And it would appear there's often sort of an empirical relationship between species' competitive abilities for nitrogen and their ability to disperse into new fields. And so one idea is that if you're a sufficiently fast colonizer, even if you're a very bad competitor, so species like these out here, you may be able to persist in the system just because you get there before other species that are better competitors are able to. Similarly, if we look at a very good competitor, something like Schizochyrium scoparium here, this is one of those late successional grasses we talked about at the beginning, we find that actually species that grow in different soil regions, that have a different rooting depth than Schizochyrium, and that grow at different times of year, so grow early in the year as opposed to the middle of the year when Schizochyrium grows, these species are able to coexist with Schizochyrium in higher abundances than other species. So here we have two different ideas of what might lead to coexistence. That you grow at a different time or depth, or simply that you move around quickly enough that you're growing in a different place. To test this, we're going to use three parameters that we hope might be proxies for these processes. So the first is R star for soil nitrate, so this is my competitive ability for nitrogen. The second is going to be Q, which is tissue nitrogen concentration, so how much of your tissue mass is made of nitrogen, and then the third is going to be your biomass in monoculture, so how much mass do you produce per unit area. And notice there's a nice conserved relationship across these three traits. If we plot them up, there's just this nice two-dimensional surface that does a pretty good job of characterizing the relationships among them. And the reason that we're choosing biomass in monoculture and tissue nitrogen concentration is that we hope that these might be some indicator of how much nitrogen a species is able to access. So Q times B star mono is the total amount of nitrogen in above ground tissues. And that might correspond to how much nitrogen you can access, both because you're able to move around a plot quickly, and also because I'm able to access nitrogen deep down in the soils or during different times of the year. And so notice this model is agnostic as to how you get more nitrogen. It merely says you might get more nitrogen through one of a series of methods. The way that this model posits coexistence among species would be to say that if there's a species here like species 3, which is a poor competitor but is able to access lots of nitrogen, it might be able to persist in mixtures with superior competitors like species 1 and 2. Because even if they take all of the nitrogen that they would get in monoculture, they leave behind a little bit of nitrogen that species 3 can access but that they can't access. The way we can write this in math is simply that my biomass in polyculture, so Bi star, is equal to my biomass in monoculture minus the sum of the total tissue nitrogen contained in superior competitors scaled by my own tissue nitrogen concentration. And if we parameterize this model here, if we just take the traits that we measured in monocultures, stick these in and use it to predict the abundances of species in the multi-species mixtures, we get this, which is not a bad fit. So here on the vertical axis, we have the observed biomass in these systems. On the horizontal axis, we have the predicted biomass. On the left is the biomass of every single species in every plot. And on the right is the biomass of the total community within each plot. And so for both of these, they sort of correspond to one another reasonably well. We explain about 20 to 25% of species level abundance and about 50% of total community abundance. And remember, we haven't tuned this model at all. These are just parameters that we've measured and stuck into a model. So this is pretty good for ecology. But remember, we hope that we can do better than this because we're going to pull information from this trade-off surface. So here we have this parameterized surface, and the species traits sort of scatter around it a little bit. And to snap these traits onto the surface, to pull information out of it, we're just going to project these traits to the nearest point on that surface. So effectively saying some of this scatter around the surface might just be measurement error attributed to more complex processes that we're not including in this model. And if we do that, we get what we have on the bottom now. So we actually get these substantially better predictions. We can now explain almost half of the variation in species level biomass and almost 60% of variation in community level biomass. And if we compare this now here, these are just comparing the absolute error, the mean absolute error for these two different types of models. Larger numbers mean more error. So you notice that we have significantly less error in the model using species snapped traits, and also our predictions from snapped traits come closer to actual observations of coexistence across these plots. That's what this bottom figure is showing. So what we can take from this is that information contained in these empirically observed trade-offs does indeed contain insight that we can use to improve our predictions. And it would suggest that the mechanisms that we've included in this model probably correspond to some of the actual mechanisms that are important for determining coexistence within this system. So we've sort of achieved our goal of being able to figure out what it is, or part of what it is, that makes this system tick. So there, you have survived all 4 chapters. And to give us a quick overview of what we did, remember for chapter 1, we introduced some pattern matching mechanisms. We then showed some potential problems with that set of pattern matching methods and demonstrated a way that we might go about solving that. We then introduced some theory and insight from trade-offs that we thought might address some remaining problems. And finally, we showed how we could apply that insight to make better predictions in real-world systems. The future steps would obviously be to try to apply this in more places and see if we can't use it to sort of figure out not just how a particular system works, but how ecosystems in general work. And that's indeed what I'm hoping to do as part of the postdoc that I'm moving on to here. Working with Stan Hartpole and Helmut Hillebrand, I'm going to be applying this both to some well-studied grassland systems in Europe and also to some aquatic systems that have been sort of very heavily experimentally worked with. So we have a good idea of what it is that drives coexistence in those systems. And the best, of course, would be to expand this out to the scale of the world, but that's a longer scale. So with that, I'd like to quickly thank lots and lots of people. I have to admit, this was probably the most stressful part of putting the talk together, because I know that I've let people off, and I'm very sorry, but tons of people were super important here. To start with, I want to thank the teachers and mentors I've had over the last 6 years, and that would include my committee and the faculty at UMM, but also, of course, Dave. And I know it's a big gauche to thank Dave right before he decides my fate, but this is the last chance I have to embarrass him publicly. So I would really like to just thank him for the last 6 years. I think in general, people know in the department, Dave has a reputation for being sort of very fiercely supportive of his grad students. And he's been, I mean, you've just been wonderful, not just in helping me figure out how to be a researcher, but Dave also pushes very hard to remind us that we're people and that life is important. And when life runs into problems, he's been very helpful in sort of reminding me to take time and pull myself out of academics. And so it's meant a lot, and it's been just a fantastic 6 years, and I really want to thank you for that. With respect to fieldwork, I owe lots of people. I'm not a born field worker, and so many people have helped me keep from killing myself and others out in the field over the last couple of years. I'd also like to thank various groups of people who have helped teach me a lot of techniques and also remind me that I should have some kind of a real life in addition to an academic life, and so that would include a large list of postdocs, most of whom are no longer postdocs. A large number of people from the Tillman Lab, and all sorts of folks from within the grad program. In particular, I'd like to thank our 2011 cohort. It's been nice that we've all stayed paced together on the same number of years. It was good to know that we're all doing either equally well or equally badly, depending on how you look at things. I also want to thank my students and interns a lot, some of whom came here. You guys have been just fantastic. You taught me how to teach, but also in the course of trying to figure out how to teach, I also had to learn a lot of ecology in the process. So it's been an honor and a privilege. I really appreciate it. I just want to thank the faculty, staff, and students at UCSB. I spent almost as much time there as I have here. It was great to always have a home base to come back to whenever I went out there. In terms of life at University of Minnesota, I of course want to thank Lisa and the staff in general for helping me make sure that I do all the things I was supposed to do in order to graduate. There are a couple of groups that have been really important for running some side projects, in particular the Sugihara Lab at UC San Diego, and a group at the Smithsonian in Panama who have just been enormously generous with time and resources and sort of helped me figure out how to run large collaborative research projects. So I really want to thank them for that. In terms of friends and family, I'd like to thank the Clarks, who will be listening at some point. Also the Thompsons, who were my sort of in parentis loco, what would it be? You had a term for that. It's been nice to have
Crazy parents.
Crazy parents. Local. It's been good to have a local group here. I really appreciate that. And it means a lot that you came, so thanks. There are various generations of roommates and road buddies and friends that have been great to have. And I also, of course, want to acknowledge a ton of funding that I've gotten from all sorts of sources. I should say, in addition to being supportive and kind, Dave also is very good in that most of the research I've done has been funded either through money he's given me or money that other people gave him. So I do want to thank those various sources. And also NSF, the LTR, and several supercomputing groups for providing resources for this. In particular, the University of Minnesota Supercomputing Institute, in that they are just remarkable at just dumping resources on grad students that normally we would have to pay for. So that's really excellent. There are a lot of other people to thank who I'm sure I've forgotten. One who I haven't forgotten is Katie. Katie has put in just an atrocious amount of emotional labor over the last 5 years, and in particular over the last 2 weeks, where I expect I was a bit more painful than usual. So thank you very much for everything. You worked super hard, both to keep me sane and to make me a slightly better person. So thanks. And that's it. Y'all are now free. And if there's time, I'd love questions, but otherwise I might just get dragged off to questioning. Anyway, thank you all.
We'll give you 5 minutes of questions.
Excellent. Thank you.
Direct.
Yeah, thanks for very interesting and stimulating talk. You've done a tremendous amount of work, very impressive. I'd like to ask you about within species variation, thinking especially about plants where sometimes size matters more than species traits. So you might have a better competitor in general terms if they are on equal circumstances, but if you happen to have a much taller plant, much bigger, it can capture more resources and is going to outcompete the other regardless of the competitive abilities or the traits that they may have, the particular traits. So I was wondering, I noticed that you work mostly with plants that are either animal or they don't live very long. And I was wondering I'm more like a forest person, a tropical forest person now. So I think of long-lived plants and in those particular circumstances, thinking about how things change from small seedlings up to large trees makes a tremendous, is very important. So I was wondering how this approach that is population-based, and I assume that it has a single value for a single species, could be implemented in cases where you need to incorporate that kind of variation, especially in size along autonomy.
So the question effectively is, what about within species trait variation, and particularly what about cases where that matters more than differences among species?
Correct.
So I should say actually the system we work in, the prairie plants, are surprisingly long-lived as well. A lot of the bunch grasses can live to be 50 or more years old. So they have quite similar lifespans to tropical trees. And because of that, we actually have had to account for within species trait variation. And it's hidden in the models. I didn't talk a lot about it, but actually in order to get good predictions from that last set of models that I showed, we had to account for the observed within species variability in traits. One thing we couldn't do because we had observations from individual monocultures was look at covariability within variation. So that would be ideally what we would want, is to have some ability to track the shared variation among traits. But I mean, I totally agree that variability within a species matters a ton. And basically the way that we capture it within this model is, in addition to having that plane that shows relationships among mean traits, we would also have a covariant structure that captured variability and associations within that. And one other thing that might help, I always use the term trait a bit too loosely. So for me, example, the height of an individual at time t would be a trait of that individual. And so I know that doesn't line up to how most people use the term trait, and we've actually gotten into some trouble with that with reviewers. So in general, I think inserting variability within species matters a ton and is pretty readily incorporated into this method. But thank you, that's a neat question.
In the spirit of this being a doctorate in philosophy, would you like to talk a little bit about your thoughts about the term mechanism, how you're using it, how that implies for evolution versus ecology versus... Yeah. It seems like you probably thought about that a lot.
So I'm a modeler, primarily, I'm afraid to say. And so for me, the definition of mechanism is pretty easy. People often talk about mechanistic models versus phenomenological models, and I find that to be a problematic dichotomy. This is what some people call mechanistic, other people will call phenomenological. It's all a bit of a mess. And so I think a good way to describe the mechanistic model to me would be the intention of the person who came up with the model. So effectively, was it my intention to try to mimic a process that I think could potentially create the patterns I see, or was my intention just to come up with a thing that looked like the thing I'm trying to match? And both of those approaches have their place, but the first of those two sort of includes an inherent hypothesis about how I think the world works. And so linear regression can be a mechanistic model if you really think that the way that the world works is there's a linear relationship between this and this. And so the way that I project that into natural communities would be to say that we never know how a real natural community works, but we can posit these sort of facsimiles, these simple sets of mechanisms, and see whether they're consistent with the patterns that we see. And so in that sense, a mechanism in the world would simply be, can I come up with some simplified idea of how the world works and use it to make a system that looks like the world that I'm trying to explain? That's a super modeler's perspective, but that I think has been a helpful way for me to think about it. Cool, thank you. So, yeah.
I'm wondering about facilitation, and so, like, especially in the dimensionality reduction, that was all competition, and it makes sense, and it's really cool. But I'm wondering if you would expect it to work at all with facilitation between species, or if you have a categorically different situation.
So facilitation doesn't exist. No, so actually we had to deal with facilitation in this model. One of the species that we included in the analysis, Kentucky bluegrass actually, does not like germinating on its own. It likes growing in a mix that's already shaded. That's actually why you usually put a shade cloth on top of Kentucky bluegrass, both because it germinates better under it and because it's able to germinate under it when other things can't. So in general, you need mixes of other species for that species to do well. And so fundamentally you could. The nice thing about these approaches is you posit a mechanism, right? And so we posited a competition mechanism, but for a system where you think that facilitation matters a lot, you could just as easily posit a mechanism where you had a positive effect of species A on species B through some sort of an intermediary. So we got lucky in that we could get away with a very simple model that just included competitive interactions. But for sure, I think including I think for me, a lot of the time, facilitation is just changing the sign on your competition model. And there are all sorts of ways that you could incorporate that into this frame.
This is your last one.
All right. Make it easy.
Well, there is none, so thank you very much.
All right.
We go to 505.
Yeah.
Is that what you're for us?